Hello friends, welcome again to my channel Asma's Botany. Okay, so today we will discuss about the topic that is the polymeric gene interaction. Okay, so polymeric gene interaction is a type of intergenic type gene interactions. Okay, and you know the term intergenic means between the two different genes. The interaction is known as the intergenic, and these two different genes means the two alleles of the two different genes can be present on the same chromosome but on different place, or on the different homologue, or on the non-homologous chromosomes also. I have already discussed in the types of gene interaction. So it is the sixth type of intergenic gene interaction, that is the polymeric. Okay, so first 5 types I have already discussed. So polymeric means, poly means many. So here many genes means 2 or more than 2 genes are involved. Okay, so and it is also known as the cumulative gene interaction. Cumulative means one is showing its character when it is mixed with another thing. You can see. So cumulative means you can understand it by this example. So first of all, in this type of gene interaction, 2 or more than 2 genes determine the single phenotype. Okay, so 2 or more than 2 genes determine the single phenotype because it is the intergenic type gene interaction. So 2 genes are involved, and it is polymeric. So in this, when dominant allele of both genes are present together. Okay, so when the dominant alleles are present together, then it shows the cumulative effect. Okay, so we can understand it by 2 or 3 examples. The first example is the pericarp color in the wheat. Okay, so it is also, we can say, the grain color in the wheat, because you know that in the wheat plant the fruit is a type of caryopsis, means the pericarp and the seed coat are joined in with each other. So the fruit is known as the caryopsis. Okay, so the pericarp color in wheat is the phenotype. This character is determined by the 2 genes, first gene and second gene. First gene have 2 alleles, C1 which is dominant, which is responsible for the red color, and the small c1 which is the recessive, which is responsible for the white color of the pericarp. And the second gene also have the 2 alleles, from which the one is dominant C2, which is responsible for the red color, and small c2 recessive allele, which is responsible for the white color of the pericarp. So this red and white color, you can see 2 genes are involved in this. Okay, so here 3 conditions can arise. So when this dominant allele, either this allele C1 or this allele C2, both are dominant, any one from this is present, not both, any one at a time is present, then the seed color, the pericarp color is a light red. Okay, when the C1 and C2 are present together, then the pericarp color is dark or deep red. So remember it, here the color is light red, but here it is the dark or deep red. So here both the dominant alleles shows the cumulative effect. When any from this one is absent, then it not shows the dark or deep red. It only shows the dark or deep red color when this 2 dominant alleles from 2 different genes are present together. Remember it. And when the 2 recessive alleles are present together, then the white pericarp is formed. Okay, so we can understand it by this cross. First of all, the parent is the homozygous. First parent has the dark red pericarp, because you can see C1 and C2 both are present. Here the white color pericarp, because small c1 and small c2 means no dominant allele is present here. Okay, then form the gametes, and after the gamete formation in the F1 generation, you can see this is the heterozygous. Okay, so here you can see dark red seeds color are produced. Why? Because this C1 and C2, both the dominant alleles of two different genes are present together. So the seeds are dark red in color. The pericarp is dark red in color. Okay, the cumulative effect is shown. Okay, so then after this we will do the selfing, then form the gametes, and after this you can form this checkerboard. In the checkerboard you can see the phenotypic ratio is 9:6:1. 9 seed pericarp is dark red, 6 light red, and 1 is white. So here you can understand it when this C1, in this you can see it is dark red. Why? Because C1 dominant allele and capital C2 both are present. Here also the dark red pericarp is present, but here you can see here light red. Why? Because both the dominant alleles which are from different genes are absent, because only one gene from one dominant allele from one gene is present. C1 is present, C2 is absent. So the light red color is produced, not dark red. Okay, it is because of the absence of the dominant allele from the other gene, the second gene. Okay, so here, and only one position when the white pericarp color seeds are produced. This, because in this no dominant allele is present. When only one dominant allele is present, like here C1 and here C2, then the pericarp color is light red. The most important thing is the phenotype is changed from the 9:3:3:1 from the dihybrid cross of the Mendel, because gene interactions are the exceptions of the Mendel's laws of inheritance. So it is 9:6:1. So 9 seeds are dark red, light red 6, and okay. So genotypes remain same. Okay. Okay, so this is the second example of the polymeric gene interaction, that is the polygenic inheritance in man. Okay, so it is seen in the skin color in humans and in the height in humans. Okay, these are the two examples, or many more like eye color, etc. So here these two are present. Okay, so you know the continuous and discontinuous variation. Before understanding this, you must understand about the continuous and discontinuous variation. So you know that in the discontinuous variations, in which every trait has the two contrasting forms of a character. So it is fixed that this thing has either this character or that character. Okay, but in the case of continuous variations, because in the continuous variations you can see very large range of characters are present. So it is not fixed that this thing has either this character or that character. Okay, so this skin color in humans and height in humans are the examples of the continuous characters or the continuous variations, in which you can see the skin color has not only two contrasting forms of characters. The skin color can be, in humans, can be negro or albino, but between two this many characters, many colors are present, like mulatto, dark than mulatto and light than mulatto. So because very large range of characters, it is known as a continuous character, continuous variations. Okay, so and it is polygenic inheritance in man is known as the quantitative. First, these two are the continuous variations. In both, continuous variations is present, and polygenic inheritance in male, this both character shows the quantitative. It is also known as a quantitative inheritance because in this the color of skin is depend upon the alleles, the dominant alleles, and the height in humans also depend upon the dominant alleles, the number of the dominant alleles. If higher number of dominant alleles are present, then the dark color is present, and if the less number dominant allele is present, then the albino, and between this albino many colors can be present. So see that. Okay, so the first example is the skin color in humans. In this you can see two genes are present. First gene have the two dominant alleles, two contrasting form of alleles. It is seen, but we will discuss that here very large range of characters in the end. So first of all, this is the dominant character. So A gene, it is a gene, dominant gene which expresses and produces the pigment. Okay, so means this is a gene. So in the molecular biology you can see this gene form a RNA and then form the protein. So the protein means the pigment structure is a protein, so it expresses and forms the protein, only the dominant allele. Okay, so pigment always formed by the dominant allele. This recessive allele don't form the pigment. Here is also the same dominant allele from the pigment, but recessive not. Okay, so then here if any genotype, you can see like this 4 alleles are present. If any genotype, one genotype, okay, when 4 dominant alleles are present, then the color of the human is negro, and if the 3 dominant alleles are present, here you can see 3 and here also 3. So here the dark color is dark than mulatto, and if the 2 dominant alleles are present, then the color of human being is mulatto. So 2 dominant alleles though, so mulatto color is produced. Only 1 dominant allele, then the color is light than mulatto. You can see here only 1 dominant allele, here also and here also. So light color is produced. Okay, and no dominant allele, then the production of albino color is only here. When no dominant allele is present, then albino color is produced. Okay, so we can form this, do this cross for this. Okay, and after this you can see in the F2 generation for checkerboard or the Punnett square, this is it. 5 types of characters are present. So there is a large range of the characters. So it is also known as the continuous variations. That's why. Okay, or it is the example of the polygenic inheritance also. Okay, so the ratio is deviate from the 9:3:3:1, the phenotypic ratio, and the ratio of the F2 generation is 1:4:6:4:1. This is because of this polygenic inheritance. Okay, so this is the exception. Or so listen carefully that modern scientists says that there are more than 2 genes that are responsible for this skin color in humans. Here you see only 2 genes, but modern scientists say that there are more than 2 genes. So because of the more than 2 genes, the phenotypic ratio of the F2 generation is 1:6:15 is in this very large range of characters can be seen. So in both large range of characters are present, but here more large range are present. Okay, so this is the example of polymeric gene interaction. Okay. Okay, so this is the example of height in humans. Okay, so in height in humans it is also a continuous variation type character because it has very large range of characters. So here you can see here when 4 dominant alleles are present in any genotype, then the person is dwarf in height, and if 3 dominant alleles are present, then the person is dwarf then medium, and 2 dominant alleles then medium size, medium height, and 1 dominant allele then tall then medium, and if the person is tall when no dominant allele is produced. So it is the genetic. Okay, so you can see the change in the phenotype of the F2 generation due to this inheritance. This is the polygenic inheritance. Okay, so the example, the name of the human which is the tallest in the world, the tallest human in the world is the Sultan Qasim, which have the height is about 8 foot 3 inches or 251 centimeters. Okay, from Turk. And the shortest human in the world is the Chandra Bahadur Dangi from Nepal, which have the height 54.6 centimeter. And in India, the shortest woman in the India is Jyoti MK from Nagpur, which has a height 63 centimeters. Okay, so this is because of this polygenic inheritance, the genetic character. Okay, so I hope you understood, and stay tuned for the next videos, and if you like the videos, press the like button and share it, and don't forget to subscribe my channel Asma's Botany and press the bell icon for the latest update. Thank you.